Many of my research questions start at the protein level, and proteins directly carry out the many functions in the cell and are modified in various ways which affect the cellular function of the protein. So my research focuses on understanding how proteins in the cell, tissue, or organism respond to different biological conditions. And specifically, we look at how mutations in proteins in development can affect the functional output of the model that we're interested in through differences in the cell's collection of proteins called the proteome. So I have the advantage of working in two research labs. Dr. Brooke McCartney's lab is a fly development lab, and Dr. Jonathan Minden's lab is a proteomics lab. To address the biological questions that I'm interested in, we're using Drosophila melanogaster, the common fruit fly, as a model, and we're also using a proteomic technique called two-dimensional gel electrophoresis, or 2D DGE. 2D DGE is a protein separation technique that allows us to compare all of the proteins between two or more samples on the same gel. And we can do this by fluorescently labeling one sample green and one sample red. We can mix those two samples and separate the proteins by their charge and molecular weight. And proteins that are similar between the samples appear as yellow, while protein differences between the samples are only shown in green and red, which provide clues for how the function of the proteins are really modified. So 2D DGE is an extremely difficult, time-consuming technique to master. To help me with these experiments, I lead a team of 4 undergrads, which allow me the flexibility to take my research where I want it, and also ask the biological questions that I feel are very interesting. So my project is looking at protein changes between normal cells versus disease cells resulting from the loss of a specific protein called APC. APC is a tumor suppressor with a diverse set of functions which have a myriad of effects on the cell. We are still trying to really understand what these functions are and what they really mean for the cell, whether it's in the normal context or a cancerous cell. Our proteomic approach revealed a new role for APC proteins in the normal developing fly embryo. We found that APC proteins regulate post-translational modifications of many types of proteins, including a diverse set of metabolic proteins. In collaboration with the University of Pittsburgh core facility, we also have identified a protein that plays a role in APC's signaling function, which we believe will help us better understand APC's role in colon cancer.